Good morning. This is my first video of my alerts publications. These are summaries in various websites about new publications, which are rather quirky and interesting, and I think somewhat informative. And let's see whether you enjoy it or not. If you go to my IVFDaily website and go to the alert section, you have to click on and pass a spider test. But there's just a list of all recent alerts that I've added to my alerts file. There's several hundred there. These are alerts that come up in my news feeds in a whole variety of websites which are involved in summarizing medical and scientific publications. There is just so much stuff being published everywhere, and a lot of journals are very hard to read. And using these alerts, such as in Medical Express, these are nice summaries of articles which add a little bit more than just the straight scientific publications. They're a great way just to catch up and learn something quirky. And I'm basically going to summarize some of these for you. Okay, let's start. So in today's alert, number one, I'm talking about sperm bots, acne drugs that restore spermatogenesis, dangers of falling birth rates overstated, donor egg births may carry more risks, imaging techniques that reveal an ecosystem that determines how eggs mature in ovaries, and DADs influence embryo growth via molecular signatures, research reveals. Once again, you can go to my IVFDaily website and select alerts. So the first one, sperm bots. This is an article published in Nature and summarized in Medical Express called Scientists Invent Sperm Bots that they piloted through a fake cervix and uterus. It's all space age stuff. But researchers have created magnetically controlled sperm bots that they can pilot around and monitor in real time. These tiny microbots are bull sperm cells coated with magnetic nanoparticles. Researchers haven't tested them yet inside real organisms, whether cow or human yet, but they demonstrated that they can control the sperm bots inside a life-size anatomical model of the female human reproductive system and track their progress with x-rays. It's all just a bit of fun, a bit of uterus, but you can see there may be some value in it. So when it comes to reproductive health, scientists say that the microbots have the potential to deliver drugs directly to the uterus, fallopian tubes, and other difficult to access parts of the female reproductive system. Such a targeted drug delivery system could improve treatments for ailments such as uterine cancer and fibroids, the study authors propose. I can actually imagine a time when these bots will be attached to an embryo and will drive the embryo to the implantation site with a whole lot of growth factors. So this is all a bit of fun, but fast forward 5 years and sperm bots may become the way to get a 100% pregnancy rate. The second one is to do for guys with acne. Yes, they were the days. This is an article published in the Journal of Assisted Reproductive and Genetics, talking about a widely used acne medication called Accutane could be used to treat male infertility, a small study hints. For men with infertility caused by extremely low sperm counts or complete lack of sperm, the only medical option is to undergo surgery to collect sperm directly from the testicles and then use for IVF. But this is a significant surgery that involves discomfort, potential risks, and impossible infection, and a prolonged recovery time. Moreover, the surgery only yields sperm about half the time. Now, researchers are exploring an alternative, isotretinoin, better known as Accutane, a drug that has been used for decades to treat severe acne. In a small study published in July in the Journal of Assisted Reproductive and Genetics, the drug stimulated sperm production in some men whose baseline had no or very few sperm in their ejaculates. The idea that a well-studied drug could stimulate spermatogenesis in men with severely impaired sperm production is exciting, well, I guess it is for some, because it opens the door to a non-surgical option for men who might otherwise require invasive testicular sperm retrieval. So in conclusion, an intratesticular retinoic acid deficiency may underlie some forms of severe male factor infertility. Treatment with isotretinoin increases sperm production in some men with non-obstructive azoospermia or cryptospermia to the point of obviating the need for testicular sperm retrieval procedures. I can imagine this may be of value since it probably would take 3 to 6 or 9 or 12 months that you may be able to do that, collect some sperm and then freeze it, and then use the frozen sperm for ICSI. The next one is just a very quick one, talking about dangers of falling birth rates in the US have been dramatically overstated. This is another conversation. Pronatalism, which I hadn't heard before, is a belief that low birth rates are a problem that must be reversed, is having its moment in the US. As demographers who study fertility, family behaviour and childbearing intentions, we can say with certainty that population decline is not imminent, inevitable and necessarily catastrophic. Just need to go back to it. In other words, there is a lot of falling birth rates. In many cases, the birth rate is less than 2. 2.1 is the rate made to maintain the population, and in many Western countries, and in fact most advanced countries, it's down by about 1.5, 1.4. Some cases down about 1.1, well below replacement rates. And whilst people think this is because of infertility, to a large extent, it is largely due to social engineering by couples electing to have fewer children and later. But that doesn't mean there is not an underlying decline in fertility, particularly with male factor infertility, and I suspect in female fertility as well. So, in one hand, it's saying there is lower birth rates, and it's largely due to social issues, but that shouldn't hide the fact that beneath that is a slowly deteriorating fertility, particularly in men, as you see with falling sperm counts. The irony is that by delaying the birth time of your first child till your mid-30s and then using IVF, you're creating an industry to solve a social problem. And some people believe this is the essential problem. Okay, getting on to which I think is probably a more important one. This was an article in Medical Express. And the authors reviewed 11 studies comparing donor egg IVF to own egg IVF and found that donor egg pregnancies had significantly higher rates of hypertensive disorder in the mother, as well as preterm birth and babies that were small for their gestational age. In a separate review focusing on preeclampsia with singleton IVF pregnancy, found the condition occurred in 11% of donor egg pregnancies compared to 3.9% of own egg pregnancies. With colleagues, these are the author of the paper, I conducted a review of 8 studies. The research suggests that gestational surrogacy using donor eggs is linked to higher rates of hypertensive disorders and gestational diabetes compared to traditional surrogacy using a woman's own eggs or previous natural pregnancies. Okay. Evidence in reciprocal IVF is more limited. Than reciprocal IVF is where mainly with lesbian couples where one woman donates eggs to the other lady who then gestates it and vice versa. A 2022 study of 21 women who underwent reciprocal IVF found that hypertensive disorder occurred in 23% compared to 12%. This is in 21 women. Compared to 12.9% of the 62 heterosexual women using their own eggs, and gestational diabetes occurred 9.5% compared to 1.6%. Crucially, there is still no strong evidence to show that fertile women carrying another woman's eggs have better outcomes than infertile women undergoing the same. But a lack of evidence is not the same as evidence of safety. In some cases, pregnancy using one's own egg may still be possible. For example, fertile women in same-sex couple may only need sperm donation to conceive naturally rather than going through IVF. Women deserve full, unbiased information about the risks. That includes knowing that carrying someone else's eggs may increase the likelihood of pregnancy complications. They can then make informed decisions about whether the potential benefits outweigh the risks. This, I think, is a sleeper issue that there's a lot of stuff published on this, and they all show the same thing, that carrying donor egg, being a recipient of donor eggs carries an increased pregnancy complication risk. And this is another one which I thought was really interesting, published in Science. Imaging techniques reveal an ecosystem that determines how eggs mature and ovary age. And it goes like this. Oocytes have species-specific gene expression patterns towards growth that converge towards maturity. Age-related transcription changes were greater in oocytes than in granulosa cells across species, although mature eggs change more in humans. The authors identified ovarian sympathetic nerves and glia axon density increased in aged ovaries, and when ablated in mice, perturbed follicular genesis. This comparative atlas defines shared and species-specific hallmarks of ovarian bust. So I'm actually only recanting text that I've taken from the articles, but the point here is that if you ablate these nerves and glia systems in the ovaries, then your impact on follicular genesis. After studying more than 100,000 mouse and human cells, they identified 11 major cell types found in the ovary, including one surprise, glia, a type of support cell typically associated with nerves and most extensively studied in the brain, were also in the ovaries. At the same time, the study revealed that sympathetic nerves, the same nerves involved in fight or flight response, formed dense networks in the ovary that became even more dense with age. When the researchers ablated these nerves in mice, the animals had more eggs in reserve, but fewer that matured, suggesting that the nerves may help decide when eggs start to grow. Together, the observations on glia and sympathetic nerves suggest a new role for the nervous system in ovarian health, and I think that's rather exciting. Another support cell called fibroblast also changed with age, triggering inflammation and scarring in the ovaries of women in their 50s, years earlier than scarring appears in organs such as in lungs and liver. All this points to a brand new line of inquiry about how nerves, blood vessels, and other cell types communicate with eggs, the author said. It tells us that ovarian aging is not just about the egg cells, about the whole ovarian ecosystem. And here's another one for the dads. Dads influence embryo growth via molecular signatures, research reveals. This is an Embryo Express, the embryo journal. To induce environmental perturbations, I love this word, prospective fathers were exposed to either non-absorbable antibiotics, disrupting the gut microbiota, or to a low-protein, high-sugar diet, in other words, a McDonald's diet. To minimise experimental variability, the analysis was performed on embryos resulting from IVF. Embryos were collected approximately 4 days after fertilisation at the blastocyst stage and individually analysed to measure differences in gene expression compared to controls, blastocysts that resulted from fathers without any treatment. The results were striking. Both environmental perturbations led to significant changes in embryonic gene expression. Disruption of the paternal gut microbiota reduced the expression of key genes involved in extra embryonic tissues, as in trophectoderm, while changes in the diet were linked to a modest developmental delay. Additionally, embryos derived from older fathers showed a stronger effect on gene expression, especially on genes involved in the immune-regulated processes, indicating that paternal age is another important factor in epigenetic inheritance. So the authors note, we have demonstrated that large-scale experiments are essential to untangle how specific environmental factors contribute to epigenetic inheritance across different genetic backgrounds, said Anna Boskovic. A tightly controlled experimental design can serve as a template for future intergenerational studies. Well, that may be so, but it's interesting how the gut bacteria, the health of the father and their eating behaviour and their weight is influencing how gene expression occurs in the embryo, and I think that is the important observation. Well, I hope you'll like my first alerts. Just to say that my name's James Danger, I'm an embryologist with 50 years experience. I'm not a medical person, but this information is not medical advice, but it may help you knowing what questions to ask about your treatment with your doctor and maybe to help in understanding the advice you receive. If you like it, click subscribe, make a comment, like, but more importantly, tell everybody else what you like. And you can support my endeavours by donating my GoFund page or subscribe to my Patreon CPD program. This is partly funded. This video is partially supported by my Third Aid website, which I've written myself. It's an online EQF program since 2003 and does performance in andrology and embryology, testing people's ability to assess embryo grades of sperm factors. You can enroll as a trainee or professional. You can do small clinic or large multi-lab clinics and use for training or competency for assessment. And that, my friend, is your lot for the week. I'd like to thank you. IVF is a personal journey that involves the action of many different people, and I acknowledge all of those. It helps us to understand all aspects of that journey to make sense of your success. I hope this information may be of value. If you like the information provided, please tell other IVF clients about this service and please subscribe. Okay, I'm off.